Welcome to the Development of B Cells module. In this talk, we're going to learn about the different stages of development that occurs in the bone marrow. We will learn about the different cell types that are formed during B cell development in the bone marrow, as well as the composition of a receptor called as the pre-B cell receptor. We will also differentiate between allelic exclusion and isotypic exclusion. During B lymphocyte development, the progenitors go through multiple stages that ultimately give rise to an immature B cell. So here we have in our figure the multipotent progenitor cell, and this cell will get signaling cues to form the common lymphoid progenitor. And then this common lymphoid progenitor will become or initiate its commitment to form a B lymphocyte and form an early pro-B cell, which will further undergo differentiation to form a late pro-B cell, which will undergo differentiation to form a pre-B cell, and finally an immature B cell will be formed. Now, as you can see in the lower portion of the figure, the different stages of B cell development are regulated by the expression of different types of proteins and transcription factors. You don't have to remember the names of the transcription factors that have been provided in the figure. However, you should know the concept that during the process of differentiation, different types of proteins and transcription factors are being expressed that promote the overall development of cells. Let us now look at the different stages of B lymphocyte development and begin with the pro-B cell. Now, during B cell development, the common lymphoid progenitor will give rise to a cell that is called as the early pro-B cell. In this early pro-B cell, we have components of the VDJ recombinase that are expressed, which include RAG1 and RAG2. Since RAG1 and RAG2 are expressed, somatic recombination can begin in these early pro-B cells. Somatic recombination begins in the heavy chain genes. And if you remember, in the heavy chain genes, the gene segments that are present are the V gene segment, the D gene segment, and the J gene segment. And thus, in the early pro-B cell, the D to J recombination occurs. And so that is what is shown in this figure in the black box, where we've had a recombination between the D gene segment and the J gene segment in the heavy chain. And we now have the combined DJ gene segment. The D to J recombination event occurs on both alleles that are present on the chromosomes, and thus we all have 2 copies of each chromosome, and hence this recombination event is happening in both copies. Once the DJ recombination occurs, the cell becomes or is called as a late pro-B cell. In the late pro-B cell, the V gene segment will be joined to the DJ gene segment, and this usually occurs in only one chromosome. So even though the DJ segments were formed in both chromosomes, the joining of the V gene segment to the DJ gene segments occurs first in only one chromosome. By joining the V to the DJ gene segment, if an intact mu chain is not made, then only the V gene segment from another chromosome will be joined to the DJ segment present on that chromosome. The cell will test for the formation of an intact mu chain, and if it is still not made, then the late pro-B cell will be eliminated. A lot of times we don't have an intact mu chain being made because of the addition and removal of nucleotides that cause a disruption in the reading frame. And hence, it is not always necessary that the joining of the V, D, and J gene segments will provide an intact B cell receptor heavy chain. And hence, the cell always has to test whether it is able to make an intact mu chain, which in this case we have the V, D, and J joined together, And through alternative splicing, the constant region of the mu gene is attached to that to form the heavy chain in that late pro-B cell. If this intact heavy chain is not made, then that late pro-B cell is eliminated or directed to undergo cell death. It is estimated that at least 45% of the pro-B cells will be eliminated because they will be unable to make an intact functional mu chain. We should remember that the enzyme terminal deoxynucleotidyl transferase is expressed in the pro-B cells, and hence, in addition to the combinatorial diversity, we are observing junctional diversity in the heavy chains. Pro-B cells that are able to make an intact mu chain has to test this heavy chain for functionality. Now at this point, no light chains have been made, and hence we can't have an intact B cell receptor. And hence, in order to compensate for the absence of the light chain, surrogate proteins called V-pre-B and lambda 5 are synthesized that will function as a light chain. Now, similar to the B cell receptor, the mu chain, the V-pre-B, and the lambda 5, as shown in the figure in the black box, they interact with each other to form what is known as a pre-B cell receptor. This pre-B cell receptor is then trafficked to the cell's surface, and along with it, it has the proteins that normally associate with the B cell receptor, which are the Ig-alpha and the Ig-beta polypeptides. Thus, the pro-B cells synthesize a pre-B cell receptor, and they will test whether this pre-B cell receptor is functional. If functional mu chains or heavy chains are made, then the amino terminal tails of the adjacent VPRB and lambda 5 are able to interact. So let us see how this actually happens. Now in the figure, the portions that they're in the black box indicate the amino terminal tails of the V-pre-B and the lambda 5. Now if the heavy chain has been made appropriately, then the V-pre-B and lambda 5 terminal tails of two adjacent pre-B cell receptors are able to interact. This interaction promotes dimerization of the pre-B cell receptor, and it results in signal transduction that involves the kinases BTK and blank. This signal transduction allows the pro-B cell to undergo changes in differentiation to become a large pre-B cell. Signaling by the pre-B cell receptor results in the inhibition of the RAG1 and RAG2 enzymes. So as we can see in the figure, the part in the black box, this is where we can see the expression of RAG1 and RAG2 that corresponds to the large pre-B cell stage. Now, the absence or reduction in expression of RAG1 and RAG2 inhibits recombination, and hence the mu chain of only one allele is expressed. We don't have the enzymes to allow somatic recombination to occur in, let's say, the other chromosome. Thus, this process or this property where we can see the heavy chain expressed from only one allele present in a chromosome is called allelic exclusion. Signaling through the pre-B cell receptor makes the developing B cell responsive to the cytokine called interleukin 7. Thus, the large pre-B cells that are formed are now responsive to interleukin 7. And interleukin 7 results in the cell division or proliferation of these large pre-B cells. Thus, when the large pre-B cells are formed, they start undergoing cell division, and the population of these cells is expanded about 30 to 60 fold. And thus now we have many large pre-B cells. It should be noted that RAG1 and RAG2 are not expressed in the large pre-B cell stage. Now the large pre-B cells are able to transition to small B cells, And as we can see in the figure, the large pre-B cell does not express RAG1 and RAG2. However, when the large pre-B cell transitions to form the small pre-B cell, the small pre-B cell is able to express the RAG proteins. Now let us see what is the outcome of this expression of RAG proteins. Now if you remember, so far in this B lymphocyte, only the heavy chain has been made. The light chain has not yet been made. When the cell is in the small pre-B cell stage, the expression of the RAG1 and RAG2 enzymes allows the formation of the light chain, and this occurs through the light chain rearrangements. As we have seen before, this occurs through the V and the J gene segments being joined together. In the case of the small pre-B cell, the recombination is going to occur only on one chromosome. Usually by this stage, the expression of the terminal deoxynucleotidyltransferase is decreased, and in some cases even stopped. And hence, when we look at the light chain sequences, the junctional diversity is not as widely observed as we see in the heavy chains. If first the VJ recombination fails to make a functional light chain, then another rearrangement is tried. And so what this is that if we look at the figure, the portions in the black box where we see there is the joining of a V gene segment and a J gene segment to form the VJ gene segment, if this rearrangement does not allow or result in a functional light chain, then what will happen is the cell will try another arrangement. For example, it will try to combine another V gene segment with another J gene segment. This whole process of light chain rearrangements first occurs in the kappa chain locus. Now, several attempts of joining the V and the J gene segments can occur in order to form a functional light chain. And the way this happens is, let us first focus on the germline sequence where we see the different V and the J gene segments. And let us say in the first round of rearrangements, the V gene segment and the J gene segment shown in the smaller black boxes are combined together to form a VJ gene segment. Now, if this gene segment is unable to form a functional light chain, or it results in a non-productive join, then what will happen is another set of V gene segments and J gene segments will then be joined together, as shown in the figure in the small black boxes. So when these two gene segments are joined together, we can see the formation of another combination of the V and the J gene segment. If this also results in a non-productive join and hence does not form a functional light chain, then we can have again another combination of somatic recombination where another V gene segment and another J gene segment will be joined together to form another combined gene segment. So in this case, we can have multiple rounds of rearrangements in the hope to form a functional light chain. If the kappa chain rearrangements do not provide a functional light chain, then the lambda chain locus will undergo rearrangement. Now the light chain shows allelic exclusion because it only allows the expression of the gene segments in one chromosome, but it also shows isotypic exclusion because it allows only the expression of either a kappa light chain or a lambda light chain. Just to reiterate and summarize, the kappa light chain locus is the one that undergoes rearrangement first, and if it fails, that is when the lambda chain locus will start undergoing rearrangements in the case of B cell development. Once a functional light chain is formed, it will pair with that heavy mu chain that was formed before, and this way we can form an intact IgM that will be expressed on the surface of the B cell. And when this happens, that is when we have the immature B cell that is formed. Now, once this B cell receptor has been formed and is expressed on the cell surface, this B cell receptor has to be tested for autoreactivity. And what this means is it has to be tested for its ability to react to self-antigens. Because we have to remember that our lymphocytes are not supposed to react to our own antigens. They must have the ability to differentiate between self and non-self. And so in the case of the immature B cells, the B cell receptor that is formed has to be tested to see if it is able to react to self-antigens. Now, the immature B cells that do not display any autoreactivity and thus do not respond to self-antigens are allowed to leave the bone marrow and they proceed and complete their development elsewhere. However, immature B cells that display autoreactivity are arrested in their development and are not allowed to develop further. Depending on the interaction of the B cell receptor to the self-antigen, there can be different fates to that immature B cell. To summarize, we learned about the different types of cells that are formed during B cell development, as well as the concept of allelic and isotypic exclusion.